Okay, so welcome to the afternoon session. We're actually now into session 2, improving resource use efficiency in soft fruit. We have 4 talks in this session, and then we'll finish off with session 3, which focuses on the soft fruit genetic improvement network. Now, for those of you who are online, just as a reminder, if you are wanting to have a look at the whole agenda for the rest of the day, you can go onto niab.com website, and if you click on events, you'll find today's event and the full program is in there. But just for this session, just to run through the 4 speakers. So we start off with Sarah Arnold again. Sarah, as you recall, is currently in Brazil. So she has recorded another presentation. We're keeping our fingers crossed that we won't have any of that interference this time. So I hope that one will work well. She's going to be talking about managing activity of pollinators in protected cropping systems. Matt Papp-Rupar will be giving us an update on his coir recycling work, which many of you have heard in the past, but he's got a little bit more news on that. And then Katia Zacharaki and Mark Else from the Crop Science and Production Systems team at Niab at East Malling will be talking about maximising yield potential with vertical farming and matching nitrogen supply to demand in strawberries. So that's the lineup for the next hour. And so our first speaker is Sarah Arnold. So this is a recorded presentation, and we'll keep our fingers crossed that we can hear her loud and clear.
So firstly, why do we care? If your soft fruit crop is under-pollinated, receives too few visits from bees and other pollinators, as the fruit starts to develop and the tissues expand in an irregular way, and this can result in misshapen fruit that are unsightly and thus unmarketable. On the flip side, if you have too many bees relative to the number of soft fruit flowers that are present on the crop, the bees drain all the nectar, get frustrated about the lack of resources, so can start to chew the stigmas, cause damage to the flowers, and this in turn can translate into misshapen fruit due to damage to those developing tissues. So there's a Goldilocks zone in the middle of the correct amount of pollination for that crop, and that's what we're aiming for. Different facilities and different facility types suffer different types of problems. So polyhouses, for example, may see patchy pollination effort, heterogeneity in the activity that you get on the crop, with some bits having high levels and other bits having low levels of pollinator activity. They may see high bee mortality depending on the cladding used, especially where the bees are struggling to orientate and getting stuck in parts of the facility away from the nest. Polytunnels have more environmental issues around temperature and humidity stress, perhaps where the pollinators will just refuse to enter some areas at certain times of day or certain parts of the season. Vertical farms have a different suite of problems, especially due to the fact that they're using artificial light. This can result in high mortality again, aggression towards workers in the facilities in some cases, which can be quite a serious consideration, and low pollination effort overall, which is not ideal for quite a high value crop and high value facility. Most of the pollination at the moment in protected cropping systems is provided by Bombus terrestris audax in the UK. It's a native subspecies, and it's a good all-rounder. You can get it from several different places. Because it can buzz pollinate, it's suitable for quite a large range of crops, including things like tomato, which not all pollinators are very good at. But other species and other options are coming through in the market. So honeybees, of course, we know about. They can be good because of the sheer numbers of them in some systems. But in enclosed systems, they sometimes don't. Red mason bees are pretty exciting. They're almost at the level of being able to scale out a large scale. Not quite there yet. They currently still have quite a narrow window of availability, so April and May. So if you have a crop that needs pollinating in August, for example, that's not so ideal, but they are very effective little pollinators within that period of availability. They transfer loads of pollen. Hoverflies are really good when they're layered on top of existing pollinators like bumblebees, and they're also commercially available pretty much year round. What we don't know so much about yet is how to optimise their performance as the sole pollinator in a system. Another option that doesn't depend on insects is air blowing, so by either a mechanical leaf blower or a large fan, agitating the air around the flowers to move the pollen around within and between flowers. And we know that some of these additional strategies can be effective because in a blackberry commercial scale facility, we did some areas where we added additional hoverflies, others where we added air blowing and looked at the impact on the fruit. And where the hoverflies were deployed, we saw a small but significant increase in druplet size. So that's plumper blackberries that look more appealing. And where we added air blowing on top of the existing Bombus terrestris pollination throughout the facility, we saw a small but significant increase in fruit length. So that's filling your punnets quicker. So there are definitely benefits from adding in more diverse different pollination options within your systems. Light can be really important in protected cropping systems. So for example, some of them exclude UV, and bees can navigate without UV, but they're slower about it, less efficient about it. So a lot of these facilities that are cutting the UV out due to the nature of their cladding may see poor performance in bee colonies in particular. And we studied this in a 3-hectare polyhouse growing blackberries at commercial scale and experiencing high bee mortality. This is currently dealt with by wrapping several sides of the facility in black Mipex. We wanted to see whether there was any alternative and also see what all of these impacts are on the pollination and on the bee health. At the moment, your best options for looking at whether pollinators are healthy and whether they're working well, you've kind of got the old school methods or the technological methods. So traditionally, you could count pollinator activity on the crop, you could look at bees moving in and out of a bumblebee hive, you could open the hive up and look inside, see if the queen's present. All of that can be done, but it's time consuming, it's labour intensive. There's a lot of technology coming through that provides round-the-clock real-time data, often streamed to an app on your phone, giving you information about things like activity on the crop, sometimes even the species of pollinator on the crop, and things like the temperature and humidity in the hive, and even the frequency of the bee buzzes in the hive. So the Hive Heart device in the top right is something that I've been working with a little bit, and it's been giving some really interesting information along those lines. We know that in honeybees, the buzz frequency is quite important because it can indicate either that the queen's died or that the colony is about to swarm. We don't know if this is important in bumblebees. So all of this may highlight developing problems before it starts to translate into an issue for the crop. Here's an example of some of that pollinator monitoring in practice. So this is a high density of monitoring devices, Agrisound Poly 3.0 in the crop, and each one is giving you a heat map of the bee activity around that point. So you see some areas do seem to suffer from a little bit of pollination deficit, even though the sensors are present and giving feedback. In this facility, we focused on these two edges where we know that the bee mortality was high, but we were able to alter the films that were hung on the wall, alter the light environment and see how that translated into impacts for bees crop. And we decided to hang different colours of film against the wall as an alternative to black Mipex. And we had a look at what the colours of those actually look like. So we looked at coloured films in pink and green. And the pink's quite interesting because it's got fluorescent particles in. And these kind of boost the blue light output. They're taking in UV, putting it out to the blue wavelength. Bees can see blue quite well, and it makes it overall look quite bright and cheerful, actually. We also looked at high diffusion. So because the bees seem to be flying towards areas that were affected by the setting sun and getting stuck there, we thought, what if you add a blurring effect so that there's no longer that point source of the setting sun? It's more of a kind of general light area. Will that reduce the amount of bee misbehaviour and reduce the amount of mortality significantly? We hung these things up in along the edge of this polyhouse in kind of 9 metre sections, and then we took the samples of the film at the end of the season and tested their appearance again. The high diffusion and the green remained fairly consistent, but with that pink film, it faded significantly. So those fluorescent particles that were making it kind of bright had almost completely faded by the end of the season. So that's a consideration. This would need changing much more frequently. So what did we find? Well, the first thing is that the pink and to some extent the green films against the walls were reducing the mortality down to the same level or even better than the black Mypex. So this was great from a point of view of reducing the number of dead bees, improving bee welfare in that respect. The high diffusion wasn't causing that reduction in bee mortality. We're still seeing a lot of dead bees against those walls, so it's not just about point source of light. The pink and to some extent the green film were associated with higher bee activity overall. So that was quite exciting. They were moving around, visiting more flowers. However, when we looked at the impact on the fruit parameters, all of the films gave the same results. So same berry mass, same drupe size, same bricks. So benefits to the bees, not significant benefits to the crop. As I said, that facility doesn't have UV. We decided to look at the same thing in conditions where UV was present to see if the bees responded the same sort of way to these different films. So this was using mini tunnels, 10 meters long, this time growing strawberry. And the roof of these either had the green film, the pink film, or a regular rain cover. And then we looked at bees in microcolonies, so a small number of bees in a little box, and we were able to get data about bee welfare using the HiveHub devices. Again, the pink film gave us good activity overall, comparable to the control where there was no kind of filtering of the light. The green saw it somewhat depressed. So pink's great for the activity here. We didn't see a lot of mortality, and it was no different between any of the conditions. So no welfare issues, but no particular benefits. And in terms of the crop performance, again, the berries were identical between all three conditions. So the pink and the green seem to be, again, broadly good for bee welfare. No particular benefits to crop production. Delving into this a little bit more with those HiveHub devices, we were able to get some temperature data inside the hive. So about 33, 34 degrees is the point at which beehives experience stress. So this is bad for the adults and particularly bad for the larvae. And it did seem like there was a slight tendency for the hives that were in tunnels that had pink and green film on them to experience fewer of those extreme stress events. And this is something that we would quite like to dig into more. Tracking it over the course of the entire cropping season then that we were focusing on, it did seem like overall the temperature inside the hives in the pink film tunnels was a little bit lower, and in the control ones was a little bit higher. So it does seem like there's a small but subtle effect on kind of hive cooling that might be happening. I mentioned earlier that buzz frequency might be important. We did have a look at this to see whether the frequency of the buzzes in any way predicted bee activity on the crop. It wasn't significant, but there's a slight hint there that maybe there's something going on that I'd love to look into a little bit more. So it may be that in the future we'll be able to monitor the frequency and the amplitude of the buzzes inside a hive to tell us whether that bee box is healthy or not. And that might in turn predict their performance on the crop. So translating this into recommendations, some soft fruit crops can experience issues with pollination, particularly in environments that have got UV blocking cladding, but it's a bit more complex than that and not always consistent. Pink coloured film does seem to have positive effects on bee behaviour, bee welfare overall. Green somewhat as well, maybe a bit less. But none of these seem to have a lot of evidence that this is translating into crop quality benefits. So the economic case for doing it or not doing it is a bit more complex, especially given that pink films fade quite quickly over the course of the season. So you need to be replacing them pretty much annually. So the pros and cons of this are more about the bees than the crop at the moment. But I'm quite excited about the technological potential to monitor hive performance so that we're swapping out hives as soon as they start to degrade and kind of sensing whether there's stresses on bees that we need to remediate. I've still got about 2.5 years left on this project, so if you're experiencing problems with pollination in your facility and you want me to come and take a look or you want to chat about it, please get in touch. I really want to solve problems that are relevant to the industry. So yeah, talk to me. Just acknowledging all the partners and funders on this project, and thank you for sitting patiently through this online talk. Bye.
Okay, thank you very much to Sarah for doing that for us. Any questions for Sarah? Michelle is in the audience, is happy to take any. If not, I just would point out that Michelle's been doing another project which is funded by the Worshipful Company of Fruiterers called Too Hot to Pollinate, where she's looking at bee stress above 33 to 34 degrees C and ways of reducing that stress. And she did some research sinking beehives into the ground to try and reduce temperatures. So all of this sort of information is regularly updated in our annual Niab Fruit Review. For those of you who are online, you can view that online on our niab.com website. If you look under Knowledge Hub, you'll find fruit, and it's all on there for you. Those of you who are here today, there are more copies of these sitting outside, so please take one away if you would like to take one, or take more for your colleagues if you'd like to take them away as well. Okay, so we'll move on to our second speaker, Matt Papp-Rupar. Matt works as a Niab pathologist in Michelle Fountain's Pest and Pathogen Ecology team and is known to most of us here. He's been talking about coir recycling and his research into that for the last 2 or 3 years at this event, and he's just going to give us an update on the latest knowledge and findings. Matt.
Yeah, thank you. Some of you have heard some parts of this. Let me see if this actually works. Oh, it does actually. Great. But now we finished this project this spring, and we have now full results together. So what is happening with coir as it is that loads of it is used because it's really good to grow strawberry and raspberry. But at least in strawberry, when you grow in bags, most of the bags are then disposed, like you see there in the picture, and then just the coir is spread out in the field. And that is a problem because you have a high upfront cost for the coir. So, and if you're just burying it in the field at the end of one season, it's a waste if you can do something else. It produces high amount of waste, which you have to do something with it. So you're spending labor on it. And there's a related carbon emissions of shipping of this coir here and then just letting it rot. So putting all together, if you consider that you have about 5,000 to 7,000 or more grow bags per hectare, and the amount of hectares that we grow in the UK, you wasting about 100 million pounds per year, and which is about 1 million cubic meters of coir across the whole country every year. So it's a huge challenge. So instead, I just realizing you can't see all the text in this version for some reason. Right, well, it's good to have me here then. So the solution for this would be to kind of close the circle, have coir from this discarded piles come back into the production, and to do that you would have to have an end-to-end process. And within this project, we combine two kind of expertise. One is an Overland, a company that is specializing in recycling, and they have a process where they can automated process for taking coir bags out of the polytunnels and then process them to remove all the plastic, remove all the roots and plants and crowns, and remove all the leaves, and they separate everything, compost everything, recycle everything they can, and they end up with a lot of coir, which then needs to be processed further to be sufficiently high quality and low risk to come into your strawberry tunnels to grow strawberry again. And how we do that, we banded together to kind of bring the aspiration of Overland, where they had hundreds and hundreds and hundreds of cubic meters of this coir left and didn't know what to do. And they brought us we were brought together in this Innovate UK Growing Canton Medway project, where my kind of if you wish, my aim of one of my goals were to evaluate what needs to be done to bring this coir back into production in terms of how to mitigate biological risks, so how to get all the pests and pathogens out of the coir and weeds, and then evaluate what has changed from year 1 to year 2 in terms of chemical and physical properties of the coir, so you can know and manage it differently if needed to. And to do that, we had to get loads of data, and the most important data from kind of commercial end, so using recycled material actually on the farm, getting feedback from the growers, measure things on the farm in large scale rather than in small scale, just here. So the first and the most important barrier that we had to cross was to ensure the end user of the coir, whether that is strawberry grower, raspberry, or any other, that there is no biological risks. So we mainly specialized on Phytophthora here and on weeds, because they're the two main management issues. So if you have Phytophthora left over from the previous year, it will have impact on the yield, and you have loads of weeds left, it will have impact on your management and yield. So we took these little pouches that you see, and we packed full of inoculum for Phytophthora or weed seeds or other things with a coir, and then we passed it through recycling system at Overland, and we did some sort of temperature and time duration analysis at Niab as well. And now, as a consequence, we know at which is the optimum or the minimum required temperature, minimum required time that the coir needs to be treated at to do it efficiently in terms of energy and cost, and to make sure that the microbes and weeds are inactivated. And because those are just the target ones that we know could be a problem, we then conducted a whole microbiome analysis as well to check all the microbes that are in virgin and recycled coir at the beginning, at the end of commercial trials. We did that over 4 trials, and we looked at the taxonomy of fungi, bacteria, and oomycetes separately, and we found that actually there is no significant differences in risk that these two coirs, virgin and recycled, pose. And that is mainly, surprisingly, because there is quite a lot of hints, for example, a lot of different oomycetes already in virgin coir that we didn't expect. So we take virgin coir, we just rehydrated it, take a sample at the beginning, you already have traces of loads of different, for example, botrytis is in there, a lot of different oomycetes such as crown rot can be found in there, and pythiums that can be. So when you compare side by side, there's no increased risk in recycled material. And then when we looked at the chemical composition, that was quite clear. All the chemistry that we looked at in recycled was different from virgin, but the levels of differences was within like manageable levels that you need. So pH was higher in recycled than in virgin, but by very little, consistently by very little. We have higher conductivity overall, but the increasing conductivity, for example, in the Everbearer, was pretty much the same across the season. So when you had to flush both virgin and recycled, and you had higher amounts of total nitrogen, phosphorus, potassium, and other things straight at the beginning of the season in the coir, in recycled coir compared to virgin. So what that means that the levels are fine. So you don't have to rinse the recycled coir, if you wish, before you use it. Actually, what you need to do is rinse less, or use less management at the beginning, because they have all the nutrients already in, but you might need different irrigation, different fertigation recipe at different times rather than the same recipe as for virgin. When we looked at it, then physical properties, which are really important, like how much air porosity and water holding capacity you have, you see that both parameters are within acceptable levels for recycled and virgin, but recycled has decreased air porosity and increased water holding capacity. So that will really hugely influence your management. So virgin coir is very difficult to waterlog things, really easy to leave them too dry. The other way around for recycled. Recycled you to waterlog quicker, and it's far better to leave it a little bit, has higher water holding capacity, and we're going to see that than in on-farm trials, where we measured all the water nutrients going in the blocks that had separate irrigation, and they were on recycle, on virgin coil, on separate irrigation. And for example, in this slide here, you see calcifarm and summerberry. We have two different trials, and we always irrigated separately. And in the first year, in 2023, on Katrina, we had very comparable yield between the two virgin and recycled material, about 1.3 per plant. But we did see 7 days earlier cropping in recycled material. And we think because you have all the nutrients present in kind of ideal amount at the beginning already, rather than in virgin where it's a lot of it was running through. And we actually saved 10% of the water and fertilizer to produce the same yield. So those plots, those tunnels that had recycled coir, they had less irrigation events, especially in really high temperature days. So during the heat wave, it was 50% less irrigation almost in recycled coir because it was all on-demand irrigated based on runoff. And none of the virgin or recycled had any pest disease and weeds issues across the whole season. So we assessed that several times. Second year, we've done a different farm, different everbearer, very similar results, really good. Yield even higher than before. And this time we didn't grow in bags, but we grow in pots or in troughs, so we can have a look at the roots. And what we saw is actually what you see at the bottom, very early on, the roots in recycled coir very uniformly grow through from top to bottom of the pot. There's loads of roots, while in virgin, which is on the right-hand side, the more orangey type, you can see most of the roots are at the bottom. So the top has less roots, probably because there's less water there, less nutrients there, everything is running through, while in recycle, you have this profile much more uniform of nutrients, water, and the consequence of that is that roots are everywhere. We had really good grower feedback from this year trial, and they are actually expanding their use of recycled coir to about 5 hectares that were grown in 2025, and they kind of goal is to be 50% recycled in the next 3 years. So that was really positive from there. And the last thing that we were kind of doing is throughout the project, we were gathering data on how much water and fuel you use for collecting the coir and bringing the coir back and processing it and steaming and drying and all that. And we put all that in a big table and collected from the literature the data on virgin, how much inputs are required for the virgin material. And if you just take face value where the virgin, you just take the costs and the environmental impact of just shipping from Sri Lanka to the UK, and then within the UK, you already save 26% total carbon emissions if you do recycling. We know I've discussed with some people in Sri Lanka and some professors from India that actually they do process in Sri Lanka already. They do processing, steaming, and drying. If you account for that as well, then you've reduced your carbon footprint by recycling coir by about 40%. So all put together, we think that recycling is much more sustainable and enabling this circular economy that is local. So you use the coir that you already have, you recycle it locally, bring it back. And if this scales up, you might have a mobile operations that might be able to come and do the whole thing on the farm, while at the moment, only the taking the coir bags out of the tunnels is done on the farm. So what we now know is that the way the overland has developed the recycling system is safe. It has low risk of pest pathogens and weeds, same as a virgin substrate, and then produce really high crop at a large scale in soft fruit. And it actually does it with less water and fertilizer use, and in some years earlier than the virgin material, and at about 25 to 40% lower emissions from the substrate compared to virgin. So what happened as a consequence of this, that it's gone from kind of idea of overland is now a product that you can call John, which I think I have email here. I don't have his email here, but call John Longley at Overland Farm or Henry, and they can offer you collection of bags, recycling and bringing back at the same price or cheaper than you could get that's their aim, as they can, you can get the virgin bag. So with that, I'm gonna stop here. I'm probably overrunning massively. And
no, you're
ask for a
perfect. Your
question.
timing is spot on. Thank you very
Great.
much indeed, Matt. Any questions for Matt? Oh, lots and lots. Okay, Adam's got the microphone. If you could just wait, we'll come to each of you in turn.
Hi, Matt.
Hey.
Question. How many times can you recycle? Like I will recycle and then I finish my season. Can I recycle that again or?
Yeah, that from what we've done, it looks like it really depends on what you start with. So when you start with good material that has a very low proportion of small particles, a lot of large ones, and if you grow a June bearer in for the first season, you can easily recycle for 2 to 3 years, 2 to 3 times if you're still growing June bearer over. If you grow everbearer and you start with a slightly poorer quality coir, you might get just one recycling that you go back to strawberry, and then you can go to a different crop that there's less fussy. With
like raspberries or something for pots.
Possibly, yeah. You could do. We have grown June bearer in 3 times recycled stuff, or material that was grown 3 times before. There was some yield penalty, but sometimes that's fine. Yeah.
Thank you.
Okay, there were a couple of other hands up, but they may have been asking the same question. But there's another one from Damien and Jonathan.
Yeah, what are the downsides? What you presented, sounds like a no-brainer. Why isn't everyone doing this?
Yeah, it's not easy to get all the bags out, and it's a kind of timing and logistics. So as Overland doing now, before you had to do everything yourself, get the bags out. The steaming and the temperature control wasn't really high throughput. So they're building now machinery that will do 200, 300 cubes an hour, where you can actually do this at scale. So one is scale, and the second was availability. It takes a lot of, so their yard, when they do just the basic recycling, removing of plastic, removing of plants, is about 250 meters long and will run for weeks on end. So it's bringing the idea on the market, and now maybe someone smaller will do like a portable version of that. But basically, it was the reluctance on trial side, on the grower trial sides, because it was done poorly few times in the past. With Overland have done trials before when they've showed really bad results with recycled stuff, and they really put a brake on investment and adoption really with that. So we had to break that barrier by showing large scale trials that were done properly.
Okay, we have to move on. Jonathan, it's a very quick one.
Yeah, just a quick one. My original question's been answered, but you've said that the quality of the material you get out of the recycling process depends on what comes in. What QC processes are there to make sure that the product that's sold is consistent and the same every time?
Yeah, so that was part of the thing that they haven't touched that much, but we kind of had to establish for them a decision tree, if you wish. So they will measure particle size and water holding capacity and stuff like that, and will batch on batch decide this is worth recycling for strawberry, and this is not worth recycling for strawberry. We're gonna sell it into amenity, or this is gonna go as a fertilizer for the. So they have to decide, and the business depends on it. So they have now if you wish, like HACCP-based system, where if you hit the threshold, you can go down that route, and if you don't hit the threshold, you can't. So that was part of the business strategy that they need to develop, really.
Matt, thank you so much indeed. We must move on. Thank you very much for that. Okay, we move on to Katia Zacharaki. Katia works in Mark Elsa's Crop Science and Production Systems team here at Niab's East Malling site. And she's been doing a lot of work over the years in total controlled environment agriculture. And she's going to talk to us about maximizing strawberry yields in these systems. Katia.
Thank you very much. First of all, I want to say thank you for saying my surname very nicely. Thank you very much. So we've been working on grow rooms and vertical farming, or TCA as we like to use these days, for quite a long time. But I know probably the feeling for many people might not be quite positive and make sense, but we have to share a few things that we think worthwhile mentioning. So you tell me afterwards what you think. So generally, as you very well know, like strawberries traditionally are grown in soil, and then probably a few decades they started actually in Europe to grow them like in tabletop, but this is not the case for America or other places. So we're a little bit advanced in this sense, and we have actually quite more high yields than other regions they say, which is for our benefit, very good. The UK has actually very good production of strawberries, very tasty strawberries that they like the cold weather we have here. Not many fruits have, but strawberries, they like it. And but what we see is actually the production of local strawberries in the UK has sort of decreased the last couple of years. And the reason for that is probably that we see lots of farmers that go out of business because of the accumulated cost of energy, labor that are going through. Some they are actually bought from bigger businesses, but some others, due to location, they don't make really sense to be consolidated with bigger farms. So we see a little bit of drop, but at the same time, we see increase of imports. Now, imports, obviously, they have like some benefit, but some negatives as well if you start talking about environmental sustainability. What we see actually is that we can actually cover the local needs with what we produce in the UK, and then we import either from the Northern Europe or we import from Egypt, Morocco, Spain, Greece, or other places like that. Personally, I cannot really taste some strawberries that they are imported during winter months. I don't know about yourself, but I would prefer to have like local UK bred actually strawberries, and ideally grown like throughout the year. And we could do that using vertical farming and TCA, but we are actually miles away from doing so because of energy cost to begin with. And maybe you are probably working with soft fruit and strawberries, you know, we do not know so many things as some new entrants in the business. They think, like, for example, investors, they might think we know everything about plant physiology, how you grow, the thresholds of photoperiod, temperature, or all those sorts of things, which we didn't need really to know when we grow outdoors in polytunnels and glass houses. But when you grow in grow rooms, TCA, you really need to understand why you choose that photoperiod and why you chose that temperature, either that is like the night time, the day time, and even the spectrum. So there are lots of unknowns at the moment, which also probably contribute with some companies going out of business. But I just wanted to mention a few companies that they have stayed or not. You might actually have come across this fancy Oishi strawberry in New York that now is on the blogs. One of the things to do when you go in New York. So what they do, they sell those strawberries. There are probably 6 or 8 on very fancy box, even better than this picture I have here, for $50. So everyone wants to try to see what this is all about. And they have chosen some Japanese cultivars that they are growing actually with quite bespoke cultivation means, with cutting away the trusses that they don't look like they're going to grow in a very substantial crop. And these they have invested, they have managed to acquire lots of investment, and they are kicking and they are fine. And the way they have done so is by niche. So they have gone niche product basically. So they are not competing for price in supermarkets, but they go direct to customers, and they have their own branding, very minimalistic and very nice branding. So that's Oise. And then at the right side, I have Agricul. So Agricul was probably the first that we know company that worked with vertical farming and strawberries in the world, let's say internationally, like commercially, selling direct to customers or some supermarkets. And they used containers around Paris actually to grow hyper-local, which is another word that we use for these terms. But their business model, because they were one of the first people trying to do so, didn't quite work out, and they went out of business. But worth to remember those two companies, to my opinion. Then there are so many other international projects, so research, but also we see technology development, and we see also people trying to grow for replicating actually Oasis business plan. There are from Barcelona, Belgium, India, Canada. Canada has actually quite a lot of these sorts of businesses, a lot, maybe 3, but for me it's a lot to have 3. And some also, they are working on the propagation. element of strawberries, not so much to supply for vertical farms, but to supply for the already existing market of strawberry propagules for growing in the ground or even in polytunnels or CA glasshouse. In the UK, there have been like various attempts for strawberries, and there are some others that I have not included in this slide, but a few to mention is Niab, that we are working probably 20, 25 years with grow rooms and strawberries. And in the last couple of years, we are focused in propagation of strawberries with a 3-year program, Innovate UK, with various collaborators. ISG as well, they're trying to go into the propagation stage of strawberries and also flex farming. I call semi-commercial probably facility, 500 square meters at Somerset, that they are actually selling their produce, not as a separate line of product, but they do sell them. So what I wanted to say is that the yield of a strawberry is actually very much dependent on how well you manage your environment, but also the actual environmental conditions. So if we say, many people, they say, what is the higher bricks cultivar, which is the one with the higher yield? And all these are, of course, like probably genetically prepositioned on the cultivar, but if you don't know what is the management, or you cannot manage well actually your environmental condition, and you don't provide the temperature, relative humidity, nutrients, and all these different things to your crop that will enhance those prepositioned genetics, then you will not actually have the outcome you want. So we need always to consider that management of the environment is very critical to see these high yielding cultivars or high bricks in cultivars and things like that. If you don't do so, no high bricks, no high yield. And I just wanted to demonstrate how this actually can be evidenced to understand the importance. Now, with a cultivar called Molling Champion, when we grew the same cultivar from the same propagator with the same management practices on a polytunnel here in East Molling, we saw actually very much different yield throughout the years. And that was because in the year 2022, we had very bright, 2020, very bright summers. And you probably remember that because we had to stay at home. So everyone enjoyed our gardens. But the following years, 2021 and 2022, we had somewhat like reduced like light quality, generally. So that led to basically reduced yield. So the same genetics with the same management, but different environment led to a different outcome. And so specifically what we see is that the average class 1 yield for the year 2020 was 1.25 kilograms per plant, while the 2021 year, that was probably down 50%. And when we take actually, okay, we say the problem here was the environment. We didn't have enough light. Light availability wasn't quite there. So let's see what happens actually in a grow room, in a TCA environment. So because everything is like ideal, we provide the ideal light quality, temperature, relative humidity, and the substrate conditions. What will happen then? So what we saw was that actually average class 1 yield for the TCA room was about 2.5 kilograms. Now we need to consider also we had probably extended cultivation because we were not limited in temperature and light, but still the 2.5 kilograms average comparing to the 1.2 the previous years is probably double, which we need to highlight here. So the genetics are there, and we were only able to see the ability of those genetics to express high yield when we had very good management of environment and temperature, and to manage basically all the managing practices. Also just to note that basically the higher yield we saw on this experiment was 3.2 kilograms per plant. So we not, and also the lowest was about 0.8 kilograms per plant. So, okay, we had the average 2.5, but we had quite spread actually high and low yield. So we were why that actually happened, because we were using like quite high quality of propagules and propagation material. So looking a little bit closer on the grow room, and with this heat map, we basically try to demonstrate the low yields with red and high yields with green. We saw that there was basically a change, differences amongst different plants, but it wasn't actually correlated with the position. So there was no what we call edge effect or anything like that. So we thought that since there was no position effect and all the environmental conditions were the same, only one thing is basically remaining that is not quite there, and that is actually the propagule quality, the quality of propagation material. So what we did, we put together basically a consortia, and we started working on propagules, how we can basically have high quality propagules with no pests and diseases. And we are currently 2 years in this project. The last year, actually, in May, this summer, we're gonna go finish this project. So we are working on producing pest and disease-free propagules that they have even more enhanced genetics or like programmed flower architecture inside the crown than actually the commercially currently propagated. And what we did, we are comparing those that we produce in vertical farming in TCA conditions with those that they are commercially propagated to see whether there is a difference. And so far we can confirm that they are like very much enhanced, and there is no pest and diseases, no crown rot, which we obviously we hope to do so. And that is a very good benefit. And as we go through the last stages of this project, we will be able to communicate with you exactly how the yield will look like and how they behaved across the cropping. So for polytunnel, glasshouse, but also vertical farming. I don't have much time, so I will rush a little bit, Scott. So generally, vertical farming, as you can see, can actually enhance the yield potential, but there are also a lot of challenges that we need to take into consideration. That can be like pollination that Sarah spoke about before, and all sorts of other things. But there are also ways that we can improve that efficiency, and that, for example, can be the regulated deficit irrigation. When it comes to water being very important, we can actually, using some techniques, that we can reduce the use of water, but at the same time without having massive yield penalties. And we actually published a paper that demonstrates, so in this graph also you can see, okay, the well water obviously had higher yield, and with quite reduced actually water in the regulated deficit irrigation, you can still have high yield, which was actually 2.3 and 2.1 kilograms for the regulated deficit irrigation, while the well water average yield was 3.3 kilograms for the cultivar Molyneux, which for us is very outstanding results. And again, demonstrating what basically the cultivars can produce. And on the same paper where you can find the light, we also did a split photo period where we were trying to basically minimize the effect of regulated deficit irrigation. And we did see actually the well water with the regulated deficit irrigation. They didn't have much difference between them. And we did think that potentially the fact that we had 2 days within 24 hours had an effect on actually minimizing these effects. Although maybe we had some pollination issues that we need to explore with Sarah probably in a new project. And finally, last slide, Scott. What we explored, which I think has application with polytunnel and glasshouse, is that actually after the flowers that were initiated during propagation, we had a little bit of dip in productivity, but after that, we had a completely linear basically yield that is very useful for productivity purposes. So again, if you can manage your environment sufficiently, you can almost have a linear productivity with an ever better like Molyneux throughout 12 months of cultivation, which we think that is the most important output out of this piece of work. And thank you very much. I don't know how much I'm over time. Any questions?
Thank you very much, Katia. we haven't got much time for questions.
Yeah.
Anything burning at this stage? If not, I think we're going to move on because Mark is going to share us results of another project. At the wet centre that was running here between 2017 and 2024, we did a lot of work looking to try and match nitrogen demand, nitrogen supply to demand with raspberry, and we got some very good results with that. And Mark is now running a project similar, looking at strawberry. So Mark's going to share his update information on that. Mark.
Great, thank you, Scott, and thank you for the invitation. And before I begin, thanks to my excellent team as well, who have done most of this work. So yes, how to match nitrogen demand with supply in strawberry. So we, by we, I probably mean scientists and growers and the industry in general, probably apply too many fertilizers and too much nitrogen. And one consequence can be a very vigorous and leafy canopy. So you see there the photograph on the left, they're Marling Ace growing in the wet centre at East Marling. And you can see they're very vigorous canopy. And that might be important in polytunnel production because often the PAR, so the light that drives photosynthesis, those PAR values are below the saturating levels of photosynthesis. And so it may be that having a good leaf area in those crops is sometimes beneficial. However, if you look at the photograph on the right, which is some, again, Marling Ace in our growth chambers, this is in a non-light limiting environment. So we really don't need all of that leaf area, we don't think. We don't want those resources being converted into leaves. We want to try and focus the allocation resources into fruit. And so as well as reducing the canopy area, of course, there are also other benefits associated with that and also opportunities to reduce emissions. And so it's important if we're going to try to target nitrogen applications more efficiently and more effectively, we do that without affecting class 1 yield and berry quality. And so I'll just highlight some of the early work we did in polytunnels and then go on to talk about some new work we're doing in TCA conditions. Now, we've probably shown this slide 27 times at least, or maybe even 28, and so I won't go into detail. What we've done is using an existing nitrogen demand model that was developed for vegetables, which is why it's called VegSyst, and we've adapted that for a range of strawberry varieties and also raspberry varieties. And so the model uses thermal time and PAR in order to be able to predict dry matter production. If we can accurately predict dry matter production, we can then calculate how much nitrogen is needed to support that dry matter production. And that's how we run the model essentially. And so by doing so, then we can manipulate the weekly or the fortnightly nitrogen inputs as well as the irrigation inputs. So this is some early work. The graphs are some early work we did with Marling Vitality. And so you'll see there the graph on the left then, that's the different nitrogen supplied per plant. The blue line is the commercial control. And this is a commercial standard fertigation formulation that the agronomy team at Berry Gardens provided with us for us at the time. And you can see there the green line then is the amount of nitrogen applied in response to using the outputs from the end demand model. So a significant reduction in the amount of nitrogen applied over the cropping season for Marling Vitality. And you can see there then in the graphs on the right-hand side, no significant effects on yield or on berry size or on average berry fresh weight. And so significant potential to reduce the nitrogen input without affecting yield. And of course, this is fairly easy to do in a June bearer. It's far more challenging with an ever bearer when we have repeat flowering. And so we continue this work with an ever bearer, again, Marling Ace. And this time we were doing this work in the growth rooms. So again, a work carried out last year. We found that we could reduce inputs of nitrogen by 52% over an 8-month cropping period. And again, there was no significant difference on class 1 yields per plant. So you can see there 2.2 kilograms and 2.1 kilograms in the respective treatments over an 8-month production period. So lots of potential then to try to reduce nitrogen inputs without impacting yield and quality. So I'll move now on to a new project. We're probably in the 4th quarter of this project. It's a FIT project. It's running for 2 years. And what we're trying to do then is to optimize a growing environment within a growth room TCA system in order to maximize yields, but also trying to reduce inputs and costs. So what we're trying to do is we're trying to combine a range of different technologies and approaches. So we're always applying the precision irrigation and fertigation system that we developed here over many years. That basically means we can limit runoff to 0.1 or 0.2%, so very controlled application of water and fertilizers. We're using the end demand model to guide our nitrogen inputs. We're using new sensors to measure NPK concentrations in real time. We're quantifying effects of treatments on fruit expansion rate using LVDTs, and of course measuring responses to these treatments in terms of leaf gas exchange. And of course photosynthesis is a key process that we're interested in. And exciting, I think, for me, is we're trying to develop and use a new clean and green CO2 capturing system. So a MOF is a metal organic framework. The idea is then we can capture CO2 from the atmosphere, bring it into the growth room, release it and pulse CO2, and then reabsorb it back in if we want to. So this is a great way to try to improve the sustainability of TCA production in terms of CO2 enrichment. So again, Marling Ace, you can see there on the left, we planted those plants on the 11th of March. First fruit began to ripen on the 28th of April. So plant establishment and growth in this environment is very quick compared to polytunnels. And you can see there then over the cropping season, which was about 23 weeks, two treatments, I should say. We had a commercial control. So that was the commercial fertilizer formulation, again, recommended to us, in this case by Steve Kember. And then we use the end demand model, of course, to reduce N inputs. And you can see there on the right-hand side, that's the cumulative class 1 yield each week during the cropping season. If we look at the total yield over that time, you can see there again, very little difference between the commercial control, which was 1.8 kilograms a plant of class 1, and the low nitrogen, which is 1.7 kilograms. So about 5% lower, but not statistically significant. No real impact on berry fresh weight, individual berry fresh weight, and no real impact on soluble solids content or bricks. What we did see, though, however, is a 13% reduction in the canopy area. So we were talking earlier about trying to optimize leaf area index for this sort of growing environment. So reducing the amount of N we supply can then reduce that canopy area, which is great for light penetration and lots of other things as well. And I guess most importantly, then we achieved a 35% saving in the amount of N we applied using the end demand model over this period of time. So all good. So then we are also interested, of course, in trying to maximize class 1 yields. And so we know that using CO2 enrichment is a great way to do this. And so in a separate growth room then, we used a CO2 enrichment strategy, in this case, enriched for 24 hours, so enriched every day essentially, and applied that and compared that to the yields from plants, similar plants growing at ambient CO2. And you can see there then a 30% increase in class 1 yield per plant was achieved using CO2 enrichment, and that mostly came about due to the higher fresh weight of individual berries. So again, that helped to improve percentage grade out. Mean bricks values were the same, but again, importantly for us, of course, enriching with CO2 not only enhances yield, but it also increases canopy area. And you'll remember from what I've said so far, of course, that we're wanting to decrease canopy area, not increase it. So very good results, but still some work to do. And you'll see there on the right-hand side, the effect of CO2 enrichment on the response of photosynthesis to increasing CO2 measured with an ergo. So good, but what we need to do is to try to work out how to maximize yield and minimize canopy area, which I'll come on to in a moment. So we measured various aspects of leaf gas exchange under the low N or elevated CO2 treatments, and you can see a summaries there. So essentially, towards the end of the treatments, photosynthesis was lowered in low N plants and raised in CO2 enriched plants, and stomata conductance again was lowered in the CO2 enrichment, which actually led to a higher water use efficiency. And all of these physiological responses are those that we would expect from plants growing in a CO2 enriched environment. And we're just looking at the data now, but we have some evidence to suggest also then that there were lower stomatal densities in the plants growing under enriched CO2, which again we might expect. So we routinely calculate KPIs, key performance indicators, to be able to compare this work with work in polytunnels or in glasshouses. And you can see there the table, I won't go through the values, but you can see there the high water use efficiency when we enrich with CO2, and you can see obviously a higher productivity in terms of nitrogen per kilograms per ton. So we're able to generate high class 1 yields, Reducing nitrogen inputs by 35% didn't reduce yields or berry quality, and we know that enriching with CO2 then can raise yields significantly due to larger berries. But as I mentioned at the beginning, what we want to do then is combined CO2 enrichment with nitrogen demand. And the idea here is then we'll apply the nitrogen demand model to try to limit canopy size, but rather than enriching with CO2 continuously, what we want to try and do is to pulse CO2 at a time when we know the photosynthetic capacity is at its most optimum. And for strawberry, even in a vertical farming scenario, you have a distinct diurnal rhythm in stomatal conductance and also in photosynthesis. And so we're going to be pulsing CO2 in the morning to try to deliver the benefits of yield, but also to try to minimize that effect on canopy growth. And we're starting that work in January time. So hopefully then we'll be able to report on those results at the next meeting. So just leaves me to say thank you to my team and to the Farm and Glasshouse team and to Steve Kember, who's visiting weekly to make sure that our experiments are in line with commercial work. And thank you to the funders, Innovate UK, and also East Marling Trust for some pump priming work, Niab too, and also the very early work in this area was supported by Berry Gardens and by Richard. So we'd like to thank them for their contribution as well. I think that's everything. Thank you.
Thank you very much indeed, Mark. You do a great job for the industry. You do a huge amount of support, and we very much appreciate it and your time. Now, I'm just going to take one question. This was one question. This is actually for Katia. I'm just going to take one question for Katia here, and it's an online one. Any problems with powdery mildew in your TCA system?
Yes, so with mulling Ace, actually, we have to say that if you control actually the relative humidity and your airflow, you can have none actually. If something goes wrong with that, then you can have lots and everywhere. And this is what I've seen. When we had not very accurate relative humidity control and we had some problems with the control relative humidity, then there was like a high occurrence and severity very high. But when there are any grow rooms or any vertical farms that you are absolutely fine with that, there is no problem at all, even when there are susceptible cultivars used. That's my experience.
Thank you, Katia.